Very good afternoon, girls.
Good afternoon, ma'am.
Good afternoon, ma'am.
Ma'am.
Good afternoon,
Okay.
ma'am.
Today we are going to discuss about beta oxidation of fatty acids. Okay?
Yes,
Okay,
ma'am.
ma'am.
Yes,
Yes.
ma'am.
What is beta oxidation of fatty acids? The breakdown of fatty acids into acetyl CoA. Okay? In this cycle, in this metabolism, we are going to discuss how our fatty acid is converted to our acetyl CoA. Uh, where where did you hear this term acetyl CoA, girls?
Pancreas.
Acetyl CoA.
Citric acid cycle.
Yes, very good. In the Krebs cycle, the first reaction starts with acetyl CoA, right? Yes or no?
Yes, ma'am.
Yeah. So if the beta oxidation happens, then only our Krebs cycle will happen because the Krebs cycle needs acetyl CoA. So beta oxidation of fatty acids is very important. In our body, beta oxidation of fatty acids is very important. So in this cycle, the fatty acid is getting converted to acetyl CoA. Acetyl CoA is needed for the Krebs cycle process. What is beta oxidation of fatty acids? The breakdown of fatty acid into acetyl CoA. The conversion of fatty acid into acetyl CoA is nothing but beta oxidation of fatty acid. In this process, what happens? Oxidation happens. Okay, oxidation of fatty acids happen. Occurs in mitochondria. Where this process usually occurs? It occurs in the mitochondria, which is a powerhouse of the cell. Process is strictly aerobic. That means we need the help of oxygen for the process to happen. Aerobic means you know, right?
Yes, ma'am.
The cycle in which oxygen is needed. So this beta oxidation needs oxygen. That's why this process is strictly aerobic. After the production of acetyl CoA, it is directly fed into the Krebs cycle. This acetyl CoA, which is formed by the beta oxidation of fatty acids. The end product of beta oxidation of fatty acid is acetyl CoA. This acetyl CoA will enter into the Krebs cycle. Okay, so for the Krebs cycle process to happen, we need acetyl CoA, which is the end product of beta oxidation of fatty acids. Now you understand the importance of beta oxidation of fatty acids, girls?
Yes, ma'am.
Yes, ma'am.
Yeah, it involves three stages. Very, very important. What are the three stages? Means first, activation of fatty acids in the cytosol. Okay, whatever the fatty acids are there in our cytosol, first they must be activated. Then they will enter into mitochondria. Okay, and mitochondria will have mitochondrial matrix. Did you remember mitochondrial
Yes,
matrix?
ma'am.
Yes, in that mitochondrial matrix, the third step, that is beta oxidation proper, will happen. Okay, the beta oxidation is divided into three stages. In the first stage, activation of fatty acids happen in the cytosol. In the second stage, transport of activated fatty acids, whatever the fatty acids which are activated are transported in the second stage, where they are transported into the
Mitochondria.
mitochondria, which is a powerhouse of the cell. And in the mitochondria, the shuttle will be there, which is known as carnitine shuttle. This word you have to underline, girls, because in this track, from this carnitine shuttle only the products will enter into mitochondrial matrix. Okay, first what happens? Activation of fatty acids. Where? In the cytosol. Cell will have cytosol, cell organelles. You know, right? Cell biology. Yes or no?
Yes, ma'am.
Yeah, inside that cell, cytosol will be there. No, there our fatty acids are activated. First step is activation. Activate after activation, they have to slowly enter into mitochondria. Okay, in the mitochondria, one special shuttle will be there. Okay, that shuttle is known as carnitine shuttle. Okay, from the carnitine shuttle, this all products should enter into matrix of mitochondria. So whole beta oxidation of fatty acids involves these three: cytosol, mitochondria, and mitochondrial matrix. First we will see detailed steps. First is activation of fatty acids. Activation of fatty acids happen in the presence of enzyme acyl CoA synthetase. Okay, first step needs an enzyme known as
Acyl CoA.
synthetase,
Synthesis.
which is nothing but fatty acid thiokinase. Fatty acid thiokinase, which requires ATP, coenzyme A, Mg, magnesium. Okay, so what we'll get after the activation of fatty acid? The products we will get are fatty acid, acyl CoA, and water. Now we will see the equation. Then you will understand. See, can you see, girls?
Yes, ma'am.
See, this is the fatty acid. First step we are discussing. This is fatty acid. RCH2CH2COO. Okay, this is our fatty acid. In the presence of enzyme thiokinase, the fatty acids are activated to acyl CoA by thiokinase. Thiokinase is also known as acyl CoA synthetase. Thiokinase is also known as acyl CoA synthetase. First step in the first reaction is this one only. Fatty acid is converted with the help of thiokinase into acyl adenylate. Okay, acyl adenylate. Here one ATP is utilized and inorganic phosphate is eliminated from the reaction. Now this acyl adenylate is converted to acyl CoA. So we have to remember fatty acid is getting converted to acyl CoA with the help of ATP, with the help of coenzyme A, with the help of thiokinase enzyme.
Ma'am, what is the use of acetyl CoA, ma'am?
See, we are discussing about beta oxidation, right? This acetyl CoA enters into carnitine shuttle. First, you have to understand the importance of beta oxidation, girls. If this beta oxidation happens, we will get a end product. What?
Acetyl CoA.
Yes, that acetyl CoA is entering into Krebs cycle. Krebs cycle is very important process. So we are discussing importance of that means step by step of beta oxidation. Each product is important. This acyl CoA will enter into another second step. 3 steps should be happen in our body. Okay?
Okay, ma'am. Thank you.
Yeah. See, fatty acids are activated to acyl CoA by thiokinase. This reaction occurs in 2 steps. Can you see 2 steps here? First step and
Yes,
Yes,
second
ma'am.
step.
ma'am.
Fatty acid react with ATP to form acyl adenylate. Fatty acid reacting with ATP to form acyl adenylate. First step clear?
Yes, ma'am.
Yes,
Here
ma'am.
AMP is formed. Adenosine monophosphate. ATP, adenosine triphosphate. PPI, inorganic phosphate. CoA means coenzyme A. Next. In the second step, this acyl adenylate is converted to acyl CoA in the presence of coenzyme A. Acyl adenylate is converted to coenzyme A. Acyl CoA, very important product formed in the first reaction. That's why the activation of fatty acid to acyl CoA. That means fatty acid is converted to acyl CoA. Fatty acid reacts with ATP to form acyl adenylate, which then combines with coenzyme A to form acyl CoA. See, did you understand second step also? How acyl adenylate is converted to acyl CoA in the presence of coenzyme A?
Yes, ma'am.
Yes,
Yeah, this
ma'am.
is the second
Yes, ma'am.
step. 2 high energy phosphates are utilized since ATP is converted to pyrophosphate. Can you see the ATP conversion to PPI?
Yes, ma'am.
Yes, ma'am.
Yes. The enzyme Inorganic pyrophosphatase hydrolyzes the PPI to phosphate. The immediate elimination of PPI makes this reaction totally irreversible. Can you see this arrow only once, right? It is not a reversible reaction. If it is reversible, there will be two arrows here.
Yes, ma'am.
So it is totally
Okay, ma'am.
irreversible reaction. It is an irreversible reaction. Whatever the PPI inorganic phosphate is eliminated, it is eliminated. Again, we can't get it back. This is an irreversible reaction. Now, did you understand the first step of beta oxidation?
Yes, ma'am.
Ma'am.
Yes, ma'am.
Now, remember girls, this acyl CoA, which is formed in the first step, is entering into mitochondria. Okay. What is the shuttle I said?
Carnitine.
Yes, carnitine shuttle. It enters into carnitine shuttle. Okay. Transport of fatty acid CoA from the cytosol. This reaction happened in cytosol. This whole, the end product of this acyl CoA from the cytosol is slowly moving into mitochondria. Transport of fatty acyl CoA from the cytosol into mitochondria. Long chain acyl CoA transferase, okay, with the special transport mechanism known as carnitine shuttle. The cytosol catalyzed carnitine acyl transferase. See, acyl CoA entering into carnitine system, that is inner mitochondrial membrane, with the help of CAT1. What is this CAT1? Carnitine acyl transferase 1. This is an enzyme which is making or which is converting our acyl CoA into acyl carnitine, and then our acyl carnitine is entering into the mitochondrial matrix. See this left part of the reaction, girls, cytosol. Our acyl CoA is formed in the cytosol. Everyone know, right?
Yes, ma'am.
This acyl CoA is converted to acyl carnitine in the presence of enzyme carnitine acyl transferase 1, CAT1. This acyl carnitine is entering into inner mitochondrial membrane, whereby it is entering into mitochondrial matrix, where our beta oxidation proper, that means third step, will happen. This is second step, girls. Did you understood?
Yes, ma'am.
Yes, ma'am.
Yeah. After entering into mitochondrial matrix, again, if you see any cycle in our body, glycolysis, Krebs cycle, and electron transport chain, everything, it should continue, no? It will stop like that after one cycle?
No,
No,
ma'am.
No, again, the end products will form the precursor, and then the cycle continues. This should be continuation in our body. No cycle will stop and take rest, right?
Yes, ma'am.
No, no.
no, ma'am.
Yeah. So,
No, ma'am.
this acyl carnitine, which is entering into mitochondrial matrix, again with the help of carnitine acyl transferase 2, is getting converted to carnitine, and this carnitine is going to cytosol, and this process will continue. Okay? But here we have to focus on acyl CoA is getting converted to acyl carnitine with the help of CAT1, carnitine acyl transferase 1, and then entering into mitochondrial matrix. So this is whole known as carnitine transport system. The acyl carnitine is transported across the membrane to mitochondrial matrix by a specific carrier protein. Did you observe in the middle, yellow color letters, carrier protein?
Yes, ma'am.
Yes,
Some
ma'am.
patients will, yeah, this acyl carnitine is slowly taken inside by this carrier protein. It will carry the acyl carnitine and it will take into the mitochondrial matrix. Okay? Carrier
ma'am.
proteins, because it is carrying, no, that's why it is known as carrier protein. It is carrying our acyl carnitine into the mitochondrial matrix, where the actual beta oxidation proper will happen. Do you understood?
Yes, ma'am.
Carnitine acyl transferase 2 converts the acyl carnitine to acyl CoA. Yeah. this acyl carnitine to acyl CoA conversion, which is happening in the mitochondrial matrix. Now the third step begins. The third step is having even more steps. That means 4 sub-steps. Okay. Third step is nothing but proper beta oxidation in the mitochondrial matrix. It is having 4 steps, girls. First step is oxidation by the FAD linked dehydrogenase, hydration by hydratase, oxidation by NAD linked dehydrogenase. Fourth step is thiolytic cleavage thiolase. Beta oxidation third step involves 4 sub-steps. What are they? First one is oxidation, second hydration, third oxidation, fourth thiolytic cleavage. Can we discuss in detail what are they?
Yes, ma'am.
Yeah. First step, conversion of palmityl CoA into trans delta square enoyl CoA. The first reaction is oxidation reaction, girls. Here conversion of acyl, that means palmitoyl CoA into enoyl CoA happens. Okay, which is an unsaturated acyl CoA. And here if you see the enzyme is acyl CoA dehydrogenase. What is the enzyme involved here?
Acyl CoA.
Acyl CoA dehydrogenase.
And the reaction happens in the presence of FAD, which is a hydrogen acceptor. Here FAD is getting converted to FADH2. Here FAD is getting converted to FADH2. Can you see?
Yes, ma'am.
Yes.
This is the first step, girls. Here example is palmitoyl CoA, which is 16 carbon. Remember, palmitoyl CoA is 16 carbon compound. Okay, this 16 carbon compound R I have represented that. Total this palmitoyl CoA structure is having 16 carbons inside it, and we have to just see the product. It is converted. This palmitoyl CoA is getting converted to enoyl CoA, trans delta square enoyl CoA, with the help of enzyme acyl CoA dehydrogenase. Because FAD is getting converted to FADH2. Is it clear?
Yes, ma'am.
Yeah, ma'am.
Yes, ma'am.
Next, this second step is hydration. First is oxidation, second is hydration. Hydration means addition of water. This trans delta square enoyl CoA in the presence of water and enzyme enoyl CoA hydratase, it is getting converted to beta hydroxy acyl CoA. Can you see how the water molecule is added in the structure?
Yes, ma'am.
In the presence of enzyme enoyl CoA hydratase, because this is a hydration reaction, right? We have to call that the enzyme which is helpful is enoyl CoA hydratase. Third step is beta hydroxy acyl CoA is getting converted to beta keto acyl CoA. In the presence of enzyme beta hydroxy acyl CoA dehydrogenase. Why dehydrogenase? Because this is an oxidation reaction. Oxidation reaction is always catalyzed by the dehydrogenases. Dehydrogenases enzymes only. We know, right?
Yes, ma'am.
Yeah, here we have to see NAD is getting converted to NADH plus H plus. That means this reaction is involving our NAD. So this reaction is NAD dependent reaction. Here NAD dependent reaction is happening in the conversion of beta hydroxy acyl CoA into beta keto acyl CoA. Can you see the changes? How our hydroxy group, that means OH group, is converted to keto group. C double bond O is a keto group. Hydroxy group means OH group, COH. OH group. So that's why the name is changed. Here in the beta hydroxy acyl CoA, hydroxy group is present. But in the beta keto acyl CoA, ketone group is present. Ketone functional group is C double bond O. Hydroxy functional group is COH. This reaction is catalyzed with the help of enzyme beta hydroxy acyl CoA dehydrogenase. Why dehydrogenase? Because this is an oxidation reaction. And
Yes, ma'am.
you know, right, what is beta? Why we call this as beta? Because the functional group attached carbon is alpha carbon, and the carbon attached to this alpha carbon, we have to call them as a beta carbon. That's why the name is beta hydroxy, beta keto. Is it clear?
Yes, ma'am.
Once again, tell na, ma'am.
Yeah. See, whatever the carbon attached to functional group, we have functional groups like acid functional group COOH, ketone functional group C double bond O, hydroxy functional group COH. Like that, we have functional groups, right? The carbon which is attached to this functional group is known as alpha carbon. Okay? And the carbon which is attached to this alpha carbon is known as beta carbon. That's why we are giving beta name. Why we are giving beta name? Because it is attached to alpha carbon. Alpha carbon is attached to functional carbon. Is it clear?
Yes, ma'am.
Yeah.
Hmm. Next is last fourth step is cleavage. Whatever the product here, it is formed, right? Beta keto acyl CoA is cleaved. Cleaved means broken down. The fourth reaction is cleavage of two carbon fragments by splitting the bond between alpha and beta carbons. Here alpha carbon and beta carbon will have one bond. That bond is cleaved or broken down. Cleavage is nothing but breakdown. Between alpha and beta carbon, the bond is broken down in the fourth step. Here, by using the enzyme thiolase, acyl CoA acetyl transferase, which is in the other form known as thiolase. This thiolase is helping in the breakdown or cleavage of beta keto acyl CoA into acyl CoA and acetyl CoA. Do you understood?
Yeah, ma'am.
I said in the first slide, palmitoyl CoA is having how many carbons?
16 carbons.
So one cycle is giving how many carbons of acetyl CoA?
6.
Observe this acetyl CoA structure and tell me how many carbons are there in the acetyl CoA.
16, ma'am.
Acetyl. See these products which are formed from the cleavage. అసిటైల్ కోఏ స్ట్రక్చర్ కి ఎన్ని కార్బన్స్ ఉన్నాయి? మనకి రెండు ప్రోడక్ట్స్ ఫామ్ అయినాయి. ఒకటి అసైల్ కోఏ విచ్ ఇస్ 14 కార్బన్, అనదర్ ఈస్ అసిటైల్ కోఏ. ఐఎమ్ ఆస్కింగ్ అసిటైల్ కోఏ ఈస్ హ్యావింగ్ హౌ మెనీ కార్బన్స్?
2 carbons.
Yes, very
3
good.
carbon.
2. Excellent, very
2
good.
carbon.
Yes. Here it is only 2, right? Acetyl CoA, 2 carbons. Acetyl, this group is acetyl group, and CoA. So acetyl CoA is having 2 carbons. Yes or no?
Yes,
Yes,
ma'am.
Yes, ma'am.
So, one cycle of palmitoyl CoA. First reaction we took palmitoyl CoA, which is a 16 carbon. So 16 carbon compound palmitoyl CoA is giving acetyl CoA, which is a 2 carbon. So this happens 7 times in our body. Beta oxidation happens how many times?
7
7
times.
7 times in our body.
So how many acetyl CoA you will get? Tell me.
2.
7.
7 plus 1 extra.
8.
8. 8 is the correct answer. Okay?
Okay,
Okay,
ma'am.
ma'am.
Whole reaction, girls. These 4 steps combinedly I have given in one slide. This is the whole reaction of beta oxidation. Okay? This you have to write in your exam if this question comes. Beta oxidation is a very important question and usually it will come in the exam. You have to practice the structure and you have to write the enzyme and product. Okay, these
Okay,
four
ma'am.
steps, I have given in one slide. Can you see this?
Yes, ma'am.
Palmityl CoA, which is a 16 carbon compound, getting converted to trans delta square enoyl CoA in the presence of enzyme acyl CoA dehydrogenase, which needs FAD. Here in the first step, FAD is getting converted to FADH2. Next, this trans delta square enoyl CoA converted to beta hydroxy acyl CoA in the presence of enoyl CoA hydratase, which is a hydration reaction. It needs the help of water. First reaction is oxidation, second hydration, third again oxidation. Okay, remember in mind, whenever oxidation reaction is there, the enzyme name will be hydrogenase, hydrogenases, like that you have to write. Hydration reaction, hydratase, and you have to observe the end of the enzyme name. You know, right?
Yes, ma'am.
Yeah.
Hydrogenation.
Hydration reactions will have enzyme name as hydratase. Oxidation reactions will have enzyme name as
Hydrogenase.
dehydrogenase. Okay, first step is oxidation. Here in the oxidation step, palmitoyl CoA is getting converted to trans delta square enoyl CoA with the help of FAD, which is converted to FADH2. Next, trans delta square enoyl CoA is getting converted to beta hydroxy acyl CoA in the presence of water. This is a hydration reaction. Enzyme enoyl CoA hydratase. This beta hydroxy acyl CoA is getting converted to beta keto acyl CoA with the help of enzyme beta hydroxy acyl CoA dehydrogenase. NAD is getting converted to NADH2. This beta keto acyl CoA is getting converted to acyl CoA and acetyl CoA, which will enter in Krebs cycle. This reaction is catalyzed by thiolase and coenzyme A. Now everyone understood?
Yes, ma'am.
Yes,
Okay,
ma'am.
ma'am.
This acetyl CoA is entering into our Krebs cycle. Acetyl CoA will enter into Krebs cycle. Now we will study energetics. Beta oxidation of palmitic acid, which is a 16 carbon compound, will undergo 7 cycles and produce 8 molecules of acetyl CoA. Okay? And in each cycle, FADH2 and NADH2 is produced. Yes or no? Here, first reaction FADH2 is produced, third reaction NADH2 is produced. FADH2 gives 2 ATP, NADH2 gives 3 ATP. So 7 cycles, 5, total 5 ATPs are produced. So 7 cycles means 5 into 7, 35 ATP. Is that clear?
Yes, ma'am.
7 cycles అవుతాయి టోటల్లీ. సో ఒక్క FADH2 2 ATP ఇస్తుంది, ఇంకొక NADH2 3 ATP ఇస్తుంది. 3 + 2 ఎంత 5. 7 cycles అవుతాయి కాబట్టి 5 into 7 35 ATPs ప్రొడ్యూస్ అవుతాయి. ఓకే,
Yes, ma'am.
నెక్స్ట్.
Okay, ma'am.
ఓకే, సి yeah. So each acetyl CoA is oxidized in the citric acid to give 12 ATP. Okay, 8 into 12, 96 ATP. 2 ATP are utilized in the activation of fatty acid, and energy gain is equal to energy produced minus energy utilized. That means 35 plus 96 minus 2, 129 ATP. Okay, girls. See, energetic of fatty acid, did you understand how 35 ATP are produced? Yes or
Yes, ma'am.
no? Yeah.
Yes, ma'am.
This 35 ATP produced plus each acetyl CoA which is oxidized in the citric acid cycle gives 12 ATP. Do you have an idea about citric acid cycle, right? Krebs cycle. It gives
Yes,
12
ma'am.
ATP, right? Acetyl CoA. Yes or
Yes,
no?
ma'am.
That,
Yes, ma'am.
yeah. 8 into 12. Why 8?
8 ATPs is
Acetyl
acetyl
CoA.
CoA.
8 acetyl CoA are formed. So 8 into 12, 96 ATP. 35 plus 96. Minus 2 because 2 ATP are utilized in the activation of fatty acid. That's why we have to minus the ATP which is utilized. Then we will get 129 ATP. Okay? Okay, girls, now you understood the beta oxidation of fatty acids?
Yes, ma'am.
Everyone.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, once again, briefly, I will explain. Beta oxidation happens in the mitochondria. It is strictly aerobic. That means it happens in the presence of oxygen. And the acetyl here, the conversion of fatty acid to acetyl CoA will happen. Why we are learning means this whole process involves the production of acetyl CoA, which is very important for our Krebs cycle. So the conversion of fatty acid to acetyl CoA happens. The beta oxidation of fatty acids involve three stages, girls. Activation of fatty acid, transport of this activated fatty acid into mitochondria, and where the beta oxidation exactly happens. Okay? First, all the fatty acids are activated in the cytosol. Okay? Next, this activated fatty acids are entering into carnitine shuttle. From the carnitine shuttle, they are entering into mitochondrial matrix. So the whole cycle which we discussed just now will happen in the mitochondrial matrix of our cell. Okay? Okay.
Okay, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Yeah. See, first is this one, activation of fatty acid. Our fatty acid is activated and getting converted to acyl CoA by thiokinase. In the second step, this acyl CoA is entering into mitochondrial matrix
Mitochondria.
with the help of carrier protein. Okay? Carrier protein. Next, in the last step, this whole reactions will happen and our palmityl CoA 16 carbon compound is getting converted to acetyl CoA.
Acetyl CoA.
That's it.
Yeah, ma'am.
Any doubts you can ask me, girls. It is clear?
Yes, ma'am. It's clear.
Okay. Thank you.
Thank you.
Thank you, ma'am.